Let's begin to understand the electron transport chain in the light reaction, which that is involved the thylakoid membrane and which that is made up of electron transport chain in the chloroplast. So the chloroplast is the photosynthetic organelle used for the light reaction and dark reaction for production of ATP as well as ATP is used for the production of glucose in the absence of light by the fixation of carbon dioxide in the Calvin cycle. So let's begin to understand the electron transport chain first, and next video we will discuss about the Calvin cycle. So this is the electron transport chain contained here is the thylakoid membrane, which that inside is the lumen and outside is the outside is the stroma, and it is the thylakoid membrane. So in this way, let's begin to understand from light reaction will be occur. Here is the photosystem 2, which that is involved for the photon 680 nanometer wavelength of the light will be absorbed. After the absorption, the primary electron acceptor will be occur. In this way, the photolysis will be occur, and this is the oxygen evolving complex, which that is evolved the oxygen from water. So, for example, let's begin to understand that two molecule of water. These two molecule of water will be split into one oxygen, which that will be move into the atmosphere, while the light will be fixed and absorb the electron accept by this photosystem 2, which photosystem 2 contain antenna complex. This antenna complex will absorb the electron, accept the electron, will move more, and ultimately the primary electron acceptor is the photosystem 2, which contain photon 680 nanometer. And in this way, the electron will be flow in the membrane of thylakoid. And here is another protein is the plastoquinine. This is absorb the proton, accept the proton and electron to reduce to the pKOH2. And again it will be cycle occur and donate the electron to the cytochrome B6F. In this way, the electron will be accepted by a cytochrome, and in this way here the proton will be removed into the lumen, while the electron will be flow further. So the concentration of the proton inside will be more, so acidity will be increased while the stroma will less. And in this way, here is the plastocyanine will accept the mobile. This is the mobile protein. A plastocyanine will be accept the electron to donate the photosystem 1, which contain a primary acceptor. The photosystem 700 will receive this electron to excite. So the primary electron excitation through light dependent, and in this way this electron will be excited to accept by a ferredoxine, and this ferredoxine is very important protein which that donate the electron to the ferredoxine NADP reductase. In this way, the proton will be accept more from the stroma. After the stroma, again more decrease of the proton in the stroma, while the lumen will increase the proton. So in this way, the proton gradients can be produced. Inside is more and outside is less. So this proton will be move out, pump out. The 4 proton pump out from ATP synthetase enzyme to produce ATP from 4.